students joined or not?
Yes, sir. Yes, sir. Joined, sir.
Okay. So I welcome all of you to the GPAT online classes organized by Andhra Pradesh State Government as well as along with Andhra Pradesh State Council of Higher Education, JNTU Anantapur and JNTU Kakinada. Now, today we will be seeing about the microbiology. So, I'm going to deal with the microbiology and biotechnology. My specialization is MPharm, Pharmaceutical Technology. I'm working with RIPER, Raghavendra Institute of Pharmaceutical Education and Research, Anandapur. Okay. Now, okay. First, before going into the topic, I'll give you the syllabus of microbiology. So it consists of Introduction to Microbiology, Microscopic and Staining Techniques, Biology of Microorganisms, Microbial Spoilage, Vaccines and sera, Fungi and Viruses, Aseptic Techniques, Sterilization and Disinfection, finally Microbial Assay. In this, the red marked topics are most important topics in terms of GPAT. So, biology of microorganism, vaccine, sera, microbial assay. So, almost most of the questions in the microbiology as well as in the microbiology portion, so they are coming mainly from these three topics only. So, for conducting this 4 days GPAT microbiology classes, I collected 1998 onwards up to 2019. Up to that, whatever the question paper came in the GPAT, so that I collected, I compiled only the microbiology and biotechnology portion. So that I'm going to, all the questions also I'll be providing to you. So based on that, the questions, how it is coming in the GPAT. So based on that, the topic has been segregated here. So this, if you focus on the three topics, almost 3 to 4 questions you can attend, which is from the microbiology. So apart from that, the green, okay, marked topics, okay, so they are less important, fewer questions are there. And in relation to microbiology, so that is the blue marked, that is still less important, and black marked, okay, that is the you have to go through that, but till now there is no that much of questions from that portions at all. Now, okay, first we will start with the introduction to microbiology. Okay, so as you know that in the world, olden days, animals and plants were there. Okay, slowly because of the invention of microscope, okay, so people come to know about the microbial species which is living in the world, which cannot be seen by the naked eyes. So slowly, because of the microscopic development and electron microscope development, so the scientists started to identify various microorganisms. So the microorganisms are the living species which cannot be seen by the naked eye, which can be seen only through the microscope, that everybody knows that. So once the microbes were identified, then the scientists started to classify the organism. So earlier, animals and plants were there. Now, this group of living species, so where to keep, okay, whether to keep in the animals or in the plants. So they didn't fall in the animal category also, they didn't fall on the plant category also. So they created separate group called as Protista. So E.H. Haeckel is a scientist who include Protista apart from the Animalia and Plantae. So living species in the world, okay, they have been classified into Protista, Animalia and Plantae. So Protista consists of bacteria that is considered as the lower protist, then fungi, algae and protozoa. Fungi, algae and protozoa, they have been considered as the higher protist. Okay, so this method of classification of microorganism were having some drawbacks. So why? Because this all the microorganism has been kept in the same group, but bacteria, fungi, algae, protozoa, all are different in their activity. Okay, for example, algae, which is living in the aquatic environment, okay, and it is multicellular in nature. Protozoa also multicellular in nature. Then fungi, it is multicellular also there, unicellular also there. Unicellular means single cells individually doing the activity. Multicellular means multi cells, different cells joined together, combinedly doing the activity. Bacteria, it is purely unicellular. So this unicellular, multicellular, and the organism are living in the different places. So, for example, algae, that is photosynthetic algae. So they mainly do the photosynthesis. Protozoa ingest the food, as like human being and animals, how they are taking the food. The protozoa will take the food like through the way of ingestion. So without any discrimination, all this microbes has been kept in the one group Protista. That is the drawback. That okay. Then viruses. That is also, as you know, that there is living and non-living that cannot be included in the any of the three categories. So that is the drawback of the E. Haeckel's classification of the living species. Then invention of electron microscope. So they invented the electron microscope. Then the scientists started to view the inner structures of the microorganism. So not only the microorganism but also the animal and plant structure also well studied. So the inner differences between the cells, okay, animal plant cells and microorganism cells has been compared. So they come to know that there is a main difference in the nucleus which contain the DNA of the cell, that is a genetic material. So there is a difference between the cells. Okay, so because of the electron microscope, one concept was confirmed that cell is the basic unit of the life. So you know that animals, plant, and higher species. So though they are having the well organized structure and organs, so you know that organ made up of tissues made up of cell. So the cells are doing the activity for the respective organs. So since the cells are considered to be the basic functional unit of the living species, then the scientists started to study about the difference between the cells. Then based on the nuclear, okay, nuclear membrane and how the nucleus is present in the cell, they categorized the living species into two categories. So that is prokaryotic and eukaryotic. So in the prokaryotic, they kept the bacteria and archaea, okay, archaea or archaea bacteria. In the eukaryotic, algae, fungi, protozoa, and animal and plants has been kept. So this system of classification started after invention of the electron microscope. Now we will see the differences between the prokaryotic and eukaryotic. So in the book, Pelzer, they have given a table, so you can refer it that also. I have given in the combined manner. So for your remembering in terms of GPAT point of view, so suppose which parts are present in the eukaryotic. Okay, so I have given in the green marked region. So green marked letters. So that is present in the eukaryotic. So in the side, so all the green marked things are absent in the prokaryotic. Then if you compare in the prokaryotic, so which are all the things present in the prokaryotic, so that is absent in the eukaryotic, that is given here. So coming back up, so what the things are present in the eukaryotic we will be saying first. So nuclear membrane. So DNA which is covered by a clear, well defined nuclear membrane in the eukaryotic, that is present in the eukaryotic cell. But in the prokaryotic, nuclear membrane is not there. The DNA is located in the middle of the prokaryotic cell, that is called as the nucleoid region. So nuclear region in the prokaryotic, it's nothing but it's a center of the cell where the DNA or genetic material of the prokaryotes located. Apart from that, eukaryotic cells contain the mitochondria, chloroplast, Golgi apparatus, endoplasmic reticulum, Rough endoplasmic reticulum, smooth endoplasmic reticulum, both are there. Okay, but these organelles are absent in the prokaryotic. Then in the eukaryotic, membrane bound vacuoles. So membrane bound vacuoles means it's nothing but, sorry. So membrane bound vacuoles are nothing but gas vacuoles. Gases are present inside the cell, but that is covered by a protein coat. But in case of prokaryotic, the membrane bound gas vacuoles are not there. So membrane bound gas vacuole is nothing but a gas covered by a protein membrane. So you know that ball, we are playing cricket by using the rubber ball. So inside the ball, the halo region will be there. So it's a tightly packed. Okay. So but inside that, gases are present. So like that, a membrane-bound gas vacuoles are present in the eukaryotic, but that is absent in the prokaryotic. Pseudopodia present in the eukaryotic, absent in the prokaryotic. Histone proteins are present in the eukaryotic, absent in the prokaryotic. Histone proteins are nothing but this protein used to fold the DNA or chromosome of the eukaryotics. So eukaryotic DNA, if you place in the thread-like structure, it is very long in length. Even a single cell DNA may goes beyond 10-meter length, but that is folded into the nucleus. That is very micron size of nucleus. Okay, nanometers, not micron, nanometer size of nanometer size of nucleus, in which that DNA is folded. So for folding the DNA, histones are the protein that is helping to help the DNA folding. So that is present in the eukaryotic, absent in the prokaryotic. Sterol present in the cell wall of the eukaryotic. Sterol is a lipid absent in the prokaryotic. Then pinocytosis, cell drinking, present in the eukaryotic, absent in the prokaryotic. So then you come to the prokaryotic, gas vacuoles. So you have to understand one thing, eukaryotic cell contain vacuoles which is covered by the protein coat. That's why it is called as the membrane bound vacuoles. But in the prokaryotic, gas vacuoles are present like a bubbles, air bubbles. They will break at any time. There is no coat at all. So simple bubbles are there. As like in the water, if a air bubble created, as like in the oil, air bubble created. So it doesn't contain any coat at all. It may break at any time. So gas vacuoles are present in the prokaryotic. Mesosomes are present in the prokaryotic. This mesosomes, okay, mesosome or mesosome. So mesosome, two types of mesosomes are there: central mesosome and then peripheral mesosomes. Central mesosome, the word itself implies that central mesosomes are located near to the center or near to the DNA of the prokaryotic cell. Peripheral mesosomes present in the near to the inside of the cell wall membrane. Okay, inside of the cell wall membrane, a bubble, okay, like structure, that is called as the uh, peripheral mesosomes. Then in the prokaryotic, storage granules are there. So prokaryotic single cellular, mainly single cellular, they will store the food for their future use. But eukaryotic cell, mostly they won't store the food at all. So their food storage system will be entirely different in the eukaryotic. But in the prokaryotic, they will be stored like a granules, okay, like a bubble-like material. Inside that, food material will be stored. Various storage granules are there. PHP granules, PHP, polyhydroxybutyrate granules present in the prokaryotic, starch granules present in the prokaryotic. Okay, then another striking feature in the prokaryotic is peptidoglycan is the material which is present in the cell wall of the prokaryotic. This peptidoglycan is absent in the eukaryotic. So this slide will give you, okay, so which are all the organs and other important things present in the eukaryotic, but that's not in the prokaryotic. Same time, which parts are present in the prokaryotic, that's not present in the eukaryotic. So that's present in this slide. So next, other differences between the prokaryotic and eukaryotic, I have listed. So if you see the size of the cell of prokaryotic, that is less than 5 micrometer size. Okay. And eukaryotic, all are more than 5 micrometer size. Okay. So the yeast cell higher than 5 micrometer, algal, mold, animal and plants, they will grow for the animal and plants will grow for the several meters, and algae also grow for the several meters also. Then coming back to the next point, DNA location. As I already discussed that nucleoid region here inside the nucleus. So in the center of the cell, nucleus, well defined nuclear membrane, eukaryotic cells are having the genetic material DNA. Apart from that, in the eukaryotic cell, mitochondrial DNA also there, chloroplast DNA also there. So DNA, genetic material, some DNA material. So it is present in the chloroplast and mitochondria, but they won't do the cell activities. They will regularize the mitochondrial activity and chloroplast activity. So the DNA present in the mitochondria and chloroplast. So that is mainly activity of the chloroplast and mitochondria. Then other differences. So the circular DNA present in the prokaryotic, linear DNA present in the eukaryotic. So one copy of the DNA, one circular DNA in the prokaryotic, eukaryotic cell will have the many copies of the DNA. Okay. Then division of the DNA, metatic division. No metatic division is present in the prokaryotic. Here, metatic nuclear division is present in the eukaryotic. Then prokaryotic DNA, that is haploid in nature. Haploid means only one copy of the gene will be present in the prokaryotic. So the DNA gene is nothing but what is meant by gene is, gene is a part of DNA which produces a single protein. Okay, so in our cell, protein is present and protein will do the many activities in the every cell. So protein serves as the receptor, protein serves as the ligand, protein serves as the enzyme, and protein will do many activities. So they'll combine with the glycoprotein, carbohydrate as a glycoprotein, protein combined with the lipoprotein, then a carbohydrate and lipid, glycolipoprotein. So all the activities of the any cell is done by the protein only. So DNA, due to transcription, translation, protein will be produced. So DNA to mRNA transcription and mRNA to protein synthesis translation. So produced protein will do the many activities inside the cell. So, for example, if you take the amylase enzyme, that is a protein which is digesting the or breaking the starch into maltose. Okay, starch will be broken into the maltose by the amylase enzyme. So, this amylase enzyme produced from the DNA of the prokaryotic. So, the part of DNA which produces the amylase, that's amylase gene. So, part of enzyme, part of a DNA which produces a single protein is called as the gene. So, for example, amylase gene, only one copy of amylase gene is present in the prokaryotic cell. If because of mutation, amylase gene is mutated, amylase production will completely stop. But in the eukaryotic, more than one gene is there. Okay, so several genes of the specific proteins are all the proteins will be there. So one gene is mutated, remaining genes will do the functions. So that is haploid genome in the prokaryotic, diploid genome in the eukaryotic, more than one gene. Then introns are absent in the prokaryotic, introns are present in the eukaryotic. So intron is nothing but intron. You remember that in the DNA, okay, genetic material, there is a region, coding region for the protein production. Some non-coding region also there. So that is called as the intron. Sorry. So introns are nothing but non-coding region of the gene. So do remember, introns are non-coding regions of the gene. So these introns are absent in the prokaryotic, introns are present in the eukaryotic. Then if you take a DNA, you know that DNA is made up of the four nucleotides. Okay, guanine, cytosine, adenine, and thymine. Guanine always pairs with the cytosine. Adenine always pairs with the thymine. So always the guanine content in any DNA that will be equal to the cytosine content. Adenine content will be equal to the thymine content. Why? Because this A joins with the T. Okay, so based on the how much amount of GC is there in the DNA, based on the how much amount of AT is there in the DNA, so DNA will be classified. So in the prokaryotic, GC content will varies from 23 to 40%. So for example, some prokaryotic contain only 23% of GC, remaining 77% of AT%. So that prokaryotic cells are called the AT DNA containing prokaryotic. They are containing 73% of AT. Some prokaryotic contain 70% of GC content. So it is called as the GC DNA in the prokaryotic. So the GC content in the prokaryotic varies from 23 to 70%. But if you see the eukaryotic, 40% GC content. So plus or minus 1 or 2% or 5% variations will be there, but only 40% of GC is present in the most of the eukaryotic. Then other differences, transcription and translation. So how transcription and translation happen in the prokaryotic and eukaryotic? So there is some differences there. In the prokaryotic, transcription and translation simultaneously happen. So DNA converted into the mRNA, immediately that mRNA simultaneously will be converted into the protein. So these two processes will happen in the cytoplasmic region, that is cytosol in the prokaryotic. But in the eukaryotic, transcription will happen in the nucleus. Inside the cell, DNA is there, but in the prokaryotic, DNA is present in the cytosol. There is no nuclear membrane. So that's why the transcription and translation happen in the cytosol only. Both will happen simultaneously. But in case of eukaryotic, the transcription, conversion of DNA to mRNA, that will happen inside the nucleus of the cell. Then the mRNA come out in the cytoplasmic region. In the cytoplasmic region, transcription mRNA will be converted to the protein. So ribosomes are located outside the nucleus of the eukaryotic. So translation will happen in the cytosol region of the eukaryotic. So transcription and translation will happen in the different, they won't happen simultaneously. mRNA production happening, right? That has to come out from the nucleus. Then after that only, that mRNA will be converted into protein. Then ribosome present in the prokaryotic, that is 70S ribosome. But in case of eukaryotic, 80S ribosome, which is arrayed in the endoplasmic reticulum membrane. Endoplasmic reticulum membrane. The 80S ribosomes of eukaryotics are located. Apart from that, mitochondria and chloroplast also contain the ribosomes. So that is 70S ribosome. So logically, generally speaking, eukaryotics contain 80S ribosome only. In the GPAT question, if it is coming, okay, about which type of ribosome is present, means you strictly write that 80S ribosome in the eukaryotic, 70S ribosome in the prokaryotic. But specifically, if they are asking mitochondria and chloroplast, then you have to write about 70S ribosome in the mitochondria and chloroplast. Then in the cytoplasmic membrane of the prokaryotic, sterols are absent. Okay, but photosynthetic bacteria and respiration doing bacteria are there. This photosynthetic bacteria as well as respiration doing bacteria, they are containing the sterols. Then in the eukaryotic, sterols are present in the cytoplasmic membrane, but they are not involved in any of the respiration or photosynthesis process in the eukaryotic. But in the prokaryotic, if the simple question is asking sterol is present in the prokaryotic or not, so no, there is no sterol in the prokaryotic, directly you can tell. But photosynthetic bacteria as well as respiration doing bacteria, they will contain the sterols. Okay. Then cell wall contain peptidoglycan, eukaryotic no peptidoglycan. Reproduction in the prokaryotic asexual binary fission, other fission method of the asexual method of the reproduction will be there in the prokaryotic. Eukaryotic, if you see, asexual method also there, and then sexual method of reproduction also there. Even other than asexual and sexual method, vegetative method of reproduction also present in the plants. So you know that organ of the plant, okay, some plants, okay, that a stem is taken and placed in the sand, so that will come bring out a tree. So that's not a asexual method, that's not a sexual method, that is called as the vegetative. Okay, so vegetative or organ, okay, other method of reproduction. So eukaryotic asexual, sexual method of reproduction they'll do. Then locomotion organelles in the prokaryotic, simple fibrils, okay, will be present here. Organized structure for the locomotion is present in the eukaryotic. Even lower eukaryotic like algae, protozoa, they will have the multiple fibers along with microtubules will be there. But in case of a prokaryotic, simple fibers will be there. In the eukaryotic, animals will have the organs for the locomotion, well-defined organs, legs are there for the locomotion. Okay. Now we'll move to the next. Then that classification of prokaryotic and eukaryotic was having some drawbacks. So what the drawback is that in the prokaryotic only bacteria has been kept, okay, remaining all the species, living species has been kept in the eukaryotic. In this photosynthetic algae also there, photosynthetic algae, absorption method of food intake. So food intake, so in the living species there are three method of food intake is present in the living species. One is absorption. So they will take they will absorb the food from the surrounding. Okay. Then photosynthesis, they will synthesize the food by using the sunlight. Ingestion. Okay. So there is a separate alimentary canal, well-developed gastrointestinal system. So food is taken through the mouth or gastrointestinal system of the system. Through that, the food will be taken. So animals, we are ingesting the food. Photosynthesis, plants will do the photosynthesis. Absorption, okay, several yeast and bacteria will do. So in the previous classification in the slide, okay, so prokaryotic contain only bacteria. So here, if you see that only bacteria and archaea has been kept here, prokaryotic, eukaryotic remaining species are kept. Then virus cannot be placed anywhere. So this is the drawback of prokaryotic, eukaryotic or nuclear membrane based classification system. Then Whitaker is the scientist who has given the five kingdom of classification of living species. It's called as the Whitaker's five kingdom of kingdom concept. So based this Whitaker five kingdom of concept is based on the three main criteria. So one is food intake, another one is whether the cell is unicellular or multicellular, and whether the cell is belonging to the prokaryotic and eukaryotic. Okay, so food intake, I already told that the three method of food intake: absorption, photosynthesis, and ingestion, unicellular and multicellular. then prokaryotic and eukaryotic. So how this is considered in the Whittaker 5 kingdom of classification is we'll be seeing. So first he kept Monera. In Monera he kept only bacteria and archaea or it is called as the eubacteria. So that has been kept in the Monera. So this Monera is unicellular, prokaryotic in nature, and food intake is absorption method will be there, photosynthesis method will be there. So Monera is unicellular, prokaryotic. Unicellular, prokaryotic. Remember, food intake is absorption and photosynthesis only there. Then second group he classified into Protista. This Protista contained various algae, mold, protozoa species and other chrysophytes, dinoflagellates, euglenites were kept in the Protista. The Protista is eukaryotic unicellular. So unicellular organisms present in the eukaryotic, they have been brought into the Protista category. So earlier in the classification, eukaryotic consists of unicellular and multicellular together, but he separated the eukaryotic unicellular and made the category into Protista. Okay, so this Protista will contain three method of food intake: absorption method of food intake, photosynthetic method of food intake, ingestion method of food intake. Absorption method of food intake, most of the Protista will do. Ingestion method, protozoa will do the ingestion method. Then photosynthetic, algae will do the photosynthetic method. So all the three method of the synthesis will be present. Food intake will be present in the Protista. Then fungi, here yeast and molds has been kept. Absorption method of food intake. So this fungi, animalia and plantae, they are mainly eukaryotic multicellular. Eukaryotic multicellular. Absorption method of food intake present in the fungi, animalia, ingestion method of food intake, plantae, photosynthesis method of uh, food intake is present. So coming back, okay, this fungi contain yeast and molds. Molds are multicellular, but yeast is single cellular and some are multicellular also there. But only unicellular yeast, okay, so that's not placed in the Protista. That is also included in the fungi. So that is the only thing you have to remember. Unicellular yeast of the unicellular eukaryotic yeast kept in the fungi. Okay, so that you have to remember. This is Whittaker 5 kingdom of classification. So there is a chance in the GPAT question. Okay, so among the following kingdom, which is having the all three mode of food intake. So Protista is the kingdom which is having the all three mode of food intake: absorption, photosynthesis and ingestion. Monera, ingestion is absent. You have to remember this critical points in terms of GPAT. Then moving into the next slide. Okay, so later on, slowly the scientist identified that DNA is the genetic material. So that is mainly the functions of the every cell. So slowly the classification system started to based on the DNA. So then next classification, classifying the living species, they started by using the genetic material, that is called as the genetic material classification. Then latest classification is that Dr. Carl Woos classification, that is called as the phylogenetic classification. That's called as the three domains of life. This is based on the rRNA cistron. Okay, so rRNA is nothing but ribosomal RNA, ribonucleic acid RNA. In the ribosome, RNA is present. Okay, the RNA present in the ribosome of the cells are analyzed. So based on that, the living species are categorized. That's why it is called as the ribosomal RNA cistern based phylogenetic classification. So from the starting of the cells, how according to the Darwin's theory, okay, slowly the cells are evolved in the world before lakhs and lakhs of years. Okay, the cells are first a single cell was evolved in the world. Slowly, the single cell started to organize. They developed a multicellular organisms. Then multicellular organisms, they organize themselves. Then bigger, okay, animal and plants are developed. So that is Darwin theory of the evaluation. So the whose concept also little based on that. In the world before several years back, there is a cell, first developed cell is called as the LUCA, last universal common ancestor, LUCA. So LUCA is the first developed cell in the world according to the Carl Hughes, that is called as the last universal common ancestor. From that, archaea bacteria developed. Okay, from this archaea bacteria, the bacteria, okay, the group of bacteria developed. Slowly, the archaebacteria evolved into eukaryotic. So, how they come to know that the rRNA present in the ribosome that is doing the specific function. So that is doing the protein function in the every cell. So it may be a microorganism, maybe a plant, or maybe an animal, or maybe fungus. Any cell will have the ribosome. The ribosome will have the rRNA. That rRNA will be helpful for the protein synthesis. So the protein all are having the 20 amino acids only. This 20 amino acids mixed together in the different combinations and various proteins are made. Okay, so this rRNA will do the peptide link formation. Remember that amino acids are linked by the uh, peptide linkage or peptide bond formation, then proteins will be made. So the peptide bond formation will be initiated by the rRNA enzyme. This is called as the ribozyme. Ribozyme is nothing but RNA doing the enzyme activity. So rRNA doing enzyme-like activity, making bond between the two amino acids. It is called as the ribozyme. So this rRNA made up of only 30 to 50 nucleotides in every ribosome that is doing the fixed function. So when they analyze the rRNA of the species, they found the similarity. So the archaebacteria, they found to contain similar rRNA. Bacteria, all are containing similar rRNA. Eukaryotic, so they are containing a sub rRNA which is little different from the bacteria and archaea, but all eukaryotic eukaryote contain same type of rRNA. So based on the rRNA, so the three domains of life classification evolved, phylogenetic classification. So archaea bacteria, this archaea, okay, this archaea contains thermophilic microorganisms and halophiles. So this archaea are living in the extreme conditions of the world. Okay, so extreme conditions of the world is nothing but, so inside the volcano, when inside the volcano, the temperature will be very higher. Okay, so inside that also microorganisms are living. Okay, in acidic environment, high acidic environment, high basic environment also microorganisms are living. Under the deep deep region of earth also microorganisms are living. Under the deep sea, high pressure will be there. Under that also microorganisms are living. Other living species cannot live in the high temperature region, high base or acidic containing region, or high pressure containing region, or the region under the earth, okay, that is very deep under the earth, the other living species cannot live. Only this archaea can live inside that. So they are living in the extreme conditions of the world. Okay, archaea bacteria living in the extreme conditions of the world. They are unicellular in nature. Then bacteria, they're living in the normal conditions of the world, normal environmental conditions of the world. Eukaryota. So that contain multicellular organisms, multicellular bacteria, that is unicellular, archaea unicellular, eukaryota, multicellular. Okay, so they are also living in the normal conditions of the world. So this is called whose classification. Okay, now we'll see the differences between the three domains of life. Okay, so there may be a GPAT question that may come in the differences between the three domains of life. Okay, so archaea and bacteria both are prokaryotic, eukaryote there, that is eukaryotic. Since archaea and bacteria both are prokaryotic, their size will be 5 micron, less than 5 micron. Eukaryote, all are higher than 5 micron. 5 micron. Then cell wall of the archaea, okay, sorry here, some mistake. Okay, bacteria contain peptidoglycan, so this has to be changed to archaea bacteria. So archaea does not contain peptidoglycan, bacteria contain peptidoglycan. Here it is wrongly mentioned, so kindly forgive me. Okay, just okay, so bacteria contain peptidoglycan, archaea does not contain peptidoglycan, eukaryota contain different polysaccharides in the cell wall. Okay, then sensitivity to the antibiotic. Okay, so here one mistake happened. Okay, so this bacteria has to be written here. So just a minute. Okay, organizer, organizer, organizer.
Hello, sir.
Just a minute. Little I'm making little change in the slide.
Okay, sir. No problem, sir.
Okay,
Okay, sir.
sorry for the disturbance. Okay.
Okay, sir.
Okay. Okay, no problem. Sorry for the disturbance. Okay, really I'm extremely sorry. Okay, so bacteria, okay, made up of peptidoglycan, and archaea does not contain peptidoglycan, and eukaryote also not contain the peptidoglycan, but the cell wall is made up of the other polysaccharides. Then bacteria can be killed by the antibiotics. So antibiotic can kill the bacteria, but eukaryotic or eukaryota, archaea cannot be killed by the antibiotic. So antibiotic sensitivity not there in the archaea as well as in the eukaryota. Then in the protein synthesis, first amino acid attached in the archaea and eukaryota, that is methionine and amino acid. But in the bacteria, formyl methionine is the amino acid which will be attached to every protein. So whenever protein synthesis happen in the bacteria, so first amino acid in the proteinuria will be formyl methionine. But in case of archaea and eukaryote, methionine is the first amino acid present in the all the proteins. Okay, but later on this amino acids will be cut and removed, that is different. But whenever a protein is produced in the bacteria, first amino acid in the protein is formyl methionine, do remember. Okay, then DNA of the bacteria, that is mostly circular in nature, archaea also circular in nature, but eukaryota that is linear in nature, and plasmids are present in the bacteria as well as in the archaea. but not in the eukaryote. Histone protein absent in the bacteria, I already told that, but archaea contain histone, eukaryote also contain histone proteins. Organelles, okay, so mitochondria, chloroplast, endoplasmic reticulum, okay, like a pinocytosis process, we already discussed in the eukaryotic. So certain organelles are absent in the prokaryotic, organelles are present in the eukaryotic. Then ribosome, bacteria and archaea contain 70s ribosome, and eukaryota contain 80s ribosome. Membrane lipids, okay, some bacteria contain lipids. That lipids are ester linked, okay, ester linked unbranched fatty acids are present in the bacteria. Okay, in the eukaryota also ester linked unbranched fatty acids are present, but archaea ether linked branched fatty acids are present. So you have to memorize these things. Okay, so lipid content in the bacteria, that is containing ester linked unbranched fatty acid. Eukaryote also same, ester linked unbranched fatty acid. But in case of archaea, it is ether linked branched fatty acid is there. Then high temperature tolerance and extreme conditions tolerance. Okay, so bacteria cannot tolerate and grow. Okay, archaea can tolerate and grow. Eukaryota cannot completely tolerate and grow. But in case of bacteria, thermophilic bacteria, they can withstand up to 65 degrees Celsius. So, but archaea bacteria, they can withstand 2000 degrees Celsius also. So organism which is withstanding in the 2000 degree of Celsius, 2000 degrees Celsius of the temperature. So how the tolerability, okay, that is amazing. Then RNA polymerase type in the bacteria. Only one type of RNA polymerase is there. Archaea and eukaryote, different, several types are there. Nuclear membrane, as we have seen, then prokaryotic nuclear membrane absent. In the bacteria and archaea, nuclear membrane is absent. But in the eukaryote, nuclear membrane is present. Now, just a minute. Now, moving to the next slide. So, now, what are the 5 major groups of microorganisms are present in the microbial world we will be seeing. So, first is that bacteria, fungi, protozoa, virus, and algae. So, these are all the 5 group of microorganisms present in the world. So, bacteria, they are single-celled, disease-causing bacterial diseases to the human, animal, and plants also there. So disease causing bacteria, okay, they are having the disease causing property. Their shape is cockeye spiral rod shape. Other shapes are also there, that is rare. And this bacteria can be grow in the laboratory or laboratory in the artificial media. This bacteria can also be used to produce the useful products for the humankind. So some bacteria used for the production of enzymes. Okay, so in the fermentation, we are using the bacteria to produce some enzymes and some proteins and other chemicals also. Then fungi, yeast and molds. Okay, so they are fungi. Yeast is unicellular, mold is multicellular. So both are causing diseases very rarely. Yeast diseases and mold diseases very rare to the human being, but they are having the capability. Example for the yeast is Saccharomyces cerevisiae. Saccharomyces cerevisiae is the organism that is used in the bread making, as well as the wine and alcohol making. So Saccharomyces cerevisiae organism, example for the unicellular yeast. Then mold, you can found the mold in the bread. So if the bread is kept for the longer period, so some black color spongy like growth. So that is mold, comes under the fungi. Okay, that is multicellular. Then apart from that, mushroom also comes under the fungi. Higher molds, mushroom, we are eating mushrooms, okay, by making curries and all. So that is also comes under the fungi. So they can also grow in the artificial media as like bacteria. Then this fungi, yeast cells are used for the alcohol production and other useful product also. Molds mainly used for the antibiotic and chemical production. Penicillium notatum is a mold used for the penicillin production. Penicillium notatum mold used for the penicillin production. Then other molds also used for the various antibiotic as well as several chemicals also produced by using the mold. So the fungi species are mainly used for producing various chemicals, okay, so which is useful in the pharmaceutical industry. Then coming back to the protozoa, okay, that is amoeba, plasmodium, and etc. They're unicellular in nature, okay, so few can grow in the media, okay, but remaining cannot grow in the media. So most of the protozoa that is intracellular in nature. So the question is asked about intracellular parasite. Virus is an intracellular parasite. Protozoa also a intracellular parasite. So virus and protozoa intracellular growth. Inside the cell only they can grow. Okay. So this protozoa ingestion mode of food intake, so they rarely causes the diseases. So you know that a plasmodium falciparum, plasmodium vivax, that is causing the malarial diseases, that belongs to the protozoa group. Okay, so only few protozoa only will cause us the human diseases. The protozoa, okay, they are saprophytic in nature. They'll eat the bacteria and lower fungi, okay, lower fungi also. So they will digest the material from the surrounding. They are saprophytes. They break the a bigger molecule into the smaller molecule, increase the fertility of the land. Bacteria also doing the same, okay, they are also breaking the bigger molecule into the smaller molecule, increases the fertility of the land. Then viruses, okay, so they can grow only inside the cell. They causes the disease in the animal, human being, plant, as well as microorganism. So viruses can cause the disease to the microorganism also. They are called as the bacteriophages. So bacteriophages viruses causes diseases in the microbes. Then animal viruses also there, bacterial, plant viruses also there, human viruses also there. Okay, then they cannot be grown in the artificial media in the laboratory. They can be grown inside the cell only. So cell cultures are used to grow the viruses. Egg is used to grow the viruses. Then algae. So that is multicellular in nature. That is growing in the aquatic environment. You might have seen in the water resources green layer, that is mainly algae only. Algae only. Okay. So that is photosynthetic in nature. Okay, that algae, okay, is a food for animals and species living in the water. Okay, animal and species living in the water rarely causes the disease to the human being. Algal diseases are very rare. This algal is used for the production of agar. Agar is a material which is used in the microbiological media. So agar is prepared, isolated from the algal species only. Then now, up to now, we have seen the introduction about the introduction to the microbiology. So in the main classification system, we have seen how the microorganisms are classified in the along with the living species that we have seen, its merits and demerits. So that points in terms of GPAT, you have to memorize the things. Now we'll move to the microscopic technique. So all of you might have studied about the structure of microscope in the microbiology subject as well as in the pharmacognosy subject also. So now I'm directly moving to the techniques used in the microbiology. So in the microbiology, okay, everywhere. So light microscope, electron microscopy through microscopic techniques are there. So first we'll see the difference between the light microscopy and electron microscopy. So light microscope uses the light, so which is of 400 nanometer to 700 nanometer wavelength. So but electron microscope uses the electron beam 1 nanometer wavelength. So lower magnification can be achieved. So the magnification means how big making the image. The smaller object can be magnified into the bigger object without any missing of the details. So with clear resolution. Okay, so this light microscopy having lower magnification, so we can magnify up to 500 to 1500 times. A small object can be magnified to 500 times bigger image or 1500 times bigger image we can generate by using the light microscopy. Why? Because this wavelength of the light is higher than the electron microscopy. So high wavelength means lesser energy, lesser wavelength. Smaller wavelength means more energy. So since more energy having electron beams are used in the electron microscopy, so we can get the high magnification up to 10 lakh times higher image, higher magnification we can produce by using the electron microscopy. Then resolution power. Now, resolution power means, so if you take a 5 MP camera, you might have seen in the mobile, if we use the 5 MP camera, that will produce the image. Have some ring effect will be there. 64 MP camera. So high resolution picture will be crystal clear. So the camera mobile phones resolution, okay, the megapixel they will be talking. So like that, light microscopy lesser resolution, electron microscopy higher resolution, crystal clear image can be generated. Then specimen preparation. So the light microscopy, we have to use the slide. We will be using slide and smear preparation, microorganism coated over the slide, placed over the slide and observed under the light microscopy. So the time period required for the preparation of the slide or preparation of the specimen to view under the microscopy, that is few minutes to few hours. But in the electron microscopy, the specimen preparation may take several days. Okay, several days. Then both live organism as well as dead organism can be observed in the light microscopy, but only dead microorganism can be observed in the electron microscopy. Do remember, this is one of the important question. So live organism can be observed only in the light microscopy. In the light microscopy also, different types of light microscopies also there, we'll see later. But commonly speaking, whether live organisms are observed in the electron microscopy and the light microscopy means only in the light microscopy live organisms can be observed. Never ever, there is no chance live organism cannot be observed in the electron microscopy. Why? Because as the organism has to be kept in the vacuum region. Electron beams are going to hit on the organism and produce the image in the electron microscopy. Automatically the cells will die, or or we ourselves prepare the cells in the dead conditions. Only dead organism can be observed under the electron microscopy. Then light microscopy in terms of cost and operation, it is less expensive, low maintenance cost. Electron microscopy high expensive and high maintenance cost. So specimen observed under the normal conditions, specimen observed under the vacuum conditions in the electron microscopy. So since the in electron microscopy, specimen observed under the vacuum conditions, the cell will morphology of the cell will changes. So original structure cannot be studied in the electron microscopy. For example, I'm telling an example. A cell placed in the dead cell also, placed in the normal environment. A cell present in the vacuum. In the vacuum, high pressure will be applied. Because of the high pressure, cell will shrink. Even dead cell also shrinking will happen or structure will changes. Okay, for example, I'm telling a rubber ball, okay, or a balloon kept in the normal environment, the same balloon kept under the pressure, rubber like structure present in the vacuum. Because of the suction of the vacuum, suction of the air, the structural changes. Okay, elasticity will increase in the rubber or balloon. So like that, cell elasticity will will be changes. So original structure of the organism cannot be studied in the electron microscopy. That is a drawback in the electron microscopy. Okay. Now we'll move to the types of the light microscopy. So in the light microscopy, four types of light microscopy is there: bright field microscopy, dark field microscopy, fluorescence microscopy, phase contrast microscopy. Okay. So in the bright field microscopy, okay, so the light which is falling on the object, then it is transmitted through the object. So transmitted light will be observed in the bright field microscopy. So dead cells, okay, dead cells can be observed in the bright field. Mainly dead cells can be observed in the bright field microscopy. Stained or unstained preparation can be observed. So both stained can also be observed, unstained can also be observed. So mainly stained preparations are observed in the bright field microscopy. It is used for the grass morphological studies. So transmitted light, okay, that is used as a transmitted light will be used to create the image of the microorganism in the bright field microscopy. The dark field microscopy, here diffracted light. Okay, so bright field microscopy, transmitted light will be used to create the image of the microorganism. Dark field microscopy, diffracted light. Okay, so here all the light will not be allowed to fall on the specimen or allowed to fall on the cell. The dark field microscopy, some slit, some preventive slit will be there. So light is coming. Okay, if a light is beam of light is coming, there is a slit which will prevent the most of the light. So some diffracted light from the slit that fall on the object. Okay, so that will create the image of the uh, microorganism. So the microorganism will be dark, will be absorbed in the dark. That's why it is a dark. Okay, it will be in the dark color. That's why it is called as the dark field microscopy. Unstained, mostly, okay, unstained microorganisms are observed under the dark field microscopy. Motility studies, movement of the microorganism can be observed clearly in the dark field microscopy. So that's not possible in the bright field fluorescence microscopy. Dark field used for the motility studies, as well as grass morphological studies also. Fluorescence microscopy, so microorganisms will be stained with the fluorescent molecule. Okay, so here also dead cells are used, dead cells are observed. Okay, so it is mainly used for the diagnostic. Okay, so diagnosis of the various diseases. So from the in the laboratory, okay, laboratory, they are isolating the patient from the microorganism, from the patients. So they are isolating the microorganism from the patient's body. That microorganisms are observed under the fluorescent microscopy by using a fluorescent dye. Then they will be easily identified. So the specific disease causing organism can be identified by using the fluorescent microscopy. So that's why it is used for the diagnostic technique in the medical field. Okay, so here uh, there is a fluorescent light. Okay, that the fluorescent dye will be coated over the cell. A specific wavelength containing light will be passed over the cell, so that fluorescence having property, having chemical substances emit the light in the different color. So the fluorescent property of the cell will be used as the base in the fluorescent microscopy. In this, antibodies, okay, also used along with the fluorescent molecule. So that is immunofluorescent microscopy, newly developed technique called as the immunofluorescent microscopy, in which antibodies are used so that specific organ of the cells can be observed very clearly, or can be coated with the dye very effectively as compared to the other method. That is called as the and immunofluorescent microscopy. Then last is that phased contrast microscopy. That is a little modification of the light microscopy in this unstained preparation. Okay, and live microorganisms are observed. So, for example, if a wave of light, what is the principle of phase contrast microscopy is, if a wave of light is passing through the one medium to another medium. For example, if you take a prism, light is passing into the prism, some refraction will occur, refracted light. So, oil is there, a light falling on the oil, entering into the oil. So from air it is entering into the oil. It's a little refraction. Okay, direction of the light will changes in the oil medium, and the light which enter into the oil medium, coming out of the oil medium, another refraction. If it is entering into atmosphere, little refraction will appear. So that refracted light will be observed under the phase contrast microscopy. This phase contrast microscopy uses the refracted, REFRACTED, refracted light. Okay, so refracted light will be observed under the phase contrast microscopy. Bright field microscopy, transmitted light. Dark field, diffraction. Diffraction is different, refraction is different. Okay, so then fluorescence. Okay, it is also emitted light observed. Okay, and among the light microscopy, which is used for the live microorganism observation. So observation of live microorganism done only on the phase contrast microscopy. Okay, so in the question paper, okay, GPAT question paper, okay, which microscopy used for the stained preparation observation, unstained preparation observation. Okay, they have asked unstained preparation observation, dark field microscopy and phase contrast microscopy, two are used. Okay, if the question is unstained, only dark field or phase contrast you have to note. Live organisms, phase contrast only. No other light microscopy can be used for the live organism observation. Live organism, phase contrast. Dead organism remain live. Rarely, okay, rarely dark field and bright field live, rarely. Sorry, bright field only dead. Bright field only dead. Dark field rarely live cells. Then we'll move to the electron microscopic technique. So what all the electron microscopic techniques are there? Before going into the electron microscopic technique, the electron is having the low penetration power. That is the drawback of the electron. So it can penetrate the cell. So surface morphology of the cell can be studied by using the electron microscopy. Internal structure can be studied by the electron microscopy. For studying the internal structure of the cells, there are different methods are there that we'll be seeing here. Okay, we'll be seeing in the techniques. So do remember, surface morphology can be studied very betterly in the electron microscopy. Why? Because its penetration power is very lesser or no penetration power for the electrons. Then the electron microscopy, there are two techniques are there. One is transmission electron microscopy, another one is scanning electron microscopy. Okay, in the scanning electron microscopy, okay, scattered secondary electrons are used to create the 3D structure of the cell or organism. So transmission electron microscopy, first you understand how it is done is, for example, okay, the light is passing, okay, so light is passing from one direction. I'm sitting here, okay, so a light, okay, a light is passing on my face, on my body. So this light will have a shadow. Okay, by using the shadow, the structure of my body can be evaluated. So in the transmission microscopy, the concept is same only. So the electron beam fall on the object, okay, object, but they cannot penetrate the object. So then the shadow will be used to create the images. So primary electrons used to create the images of the cell. So 2D structures can be generated. Then in the electron microscopy, it is 360 degree. So for example, all the directions, okay, this direction they are passing the light, this direction they are passing the light, behind they are passing the light, all the shadows are captured, all will be combined to create the whole 3D structure of the human body. So just like that, the word scanning electron microscopy, okay, scanning will be done on the 360-degree of the cell, then image will be created based on that. So 3D structure will be created. So this is scanning electron microscopy. In transmission electron microscopy, different types are there. Shadow casting, ultra thin sectioning, freeze etching, technique, negative staining technique, and other methods also there. So mainly shadow casting, ultra thin sectioning, freeze etching, negative staining. So shadow casting is nothing but, okay, electron beam, that is platinum electron beam. So platinum metal will be allowed to pass from one direction. So for example, I am sitting here, okay, a dust material falling on my body. So I'm still sitting like this, dust material falling on my body, my shadow will be generated in the bottom. In the shadow, there won't be any dust material. So like that, electron falling on the object, shadow of the object will be in the downside. So that shadow will be used to create the image of the object. That's why it is called as the shadow casting. In this, platinum metal will be allowed to fall on the object. Then the shadow where the platinum metals are not present in the bottom side, that is used to create the image of the object. Then the next is that negative staining. We'll see ultra thin sectioning and freeze etching later. Negative staining. In the negative staining, object coated with a phototungstic acid. Okay, this phototungstic acid will not allow the electron to penetrate very, it will reflect the electron. Uh, for example, in a body, okay, to show the body, okay, in some functions or temple functions, they will apply some silver color, okay, silver color paint, which will be glowing. They will be applying over the body, so that their body will be seen very effectively. As like in the some bodybuilding competition. So bodybuilders will apply some glittering paints or some creams over the body that will show the structure and the cervixes. Bend and curves will be shown very nicely. Okay, so like that, photo passport tungstic acid, not photo. Okay, so passport is missing. So sorry, here passport tungstic acid. So I'll correct this slide and give it to you. So passport tungstic acid will be coated over the object, and image will be generated. That is called as the negative staining. So this is extremely useful to study about the minute structure of the bacteria. So flagella, pili, like smaller structure of the bacteria can be effectively absorbed. So virus also can be absorbed very effectively in the negative staining technique. So virus, flagella and pili can be effectively absorbed in the negative staining. Then ultra thin sectioning. Okay, this is modified technique. So just like that, just like that, take a bread. Okay, inside the bread, yeah, plum. Okay, plum fruit is there inside the bread. bread is cut into a pieces. Okay, we are taking a bread cut into pieces. Okay, so if a blum piece is there while cutting, okay, blum also cut into pieces. Okay, if you take a piece and view, you can see the structure of the blum. Okay, so combine all the pieces, okay, 3 or 4 pieces which is adjacent to that, then you can create the structure of the whole plum. Okay, the plum fruit embedded in the bread, a plum fruit embedded in the bread, cut into slices, then every slice will be observed, so you can see the structure of the plum fruit. So like that, cells are embedded in the plastic like material. The plastics are cut into 60 slices. Okay, then each slice will be absorbed separately, so that internal organ structure will be established, so that cell can be studied clearly. So you might have seen MRI scan in the head. MRI scan, so one section of the head will be taken first. One section will be taken, another section, third section, 4, 5 section of the head will be taken, MRI scan. All scans will be put together, a head, so whole brain can be generated. So like that, cell embedded in a plastic like material, a round cell embedded in a plastic material, cut into thin slices. Each slice will be taken and studied about the structure of the embedded cell. This is ultra thin sectioning. Okay, here plastic or chemical material used to create the embedding the cells. Then freeze etching. Okay, in the ultra thin sectioning, we are using plastic or chemical material to embed the cells, so that what will happen is nature of the cell will be get affected. So you melt the plastic, mix the cell inside, then cut by using the finer laser like material, so we'll be generating the 60 micrometer slices of the plastic, then observed under the microscope. Okay, so the heated plastic will change the structure of the bacteria, so that is the drawback. To avoid that, okay, usage of chemicals, freeze etching method has been developed. In the freeze etching method, okay, the cells are frozen. Okay, and frozen cells are cut into 60 micrometer slices, then they will be observed under the electron microscopy. Okay, this is the freeze etching. Then other electron microscopic technique, localization of cell constituents. So we can in the electron microscopy, we can some chemicals where it is located inside the cell, that also can be done in the electron microscopy. Then localization of enzyme, we can locate the enzyme, okay, amylase enzyme where it is located in the cell. Okay, then a protease enzyme where it is located exactly in the cell. Okay, so every enzyme where it is located inside the cell, that can be better studied by using the electron microscopy. That technique is called as the localization of enzymes. Then autoradiography, so radioisotopes are used. Okay, so this radioisotope go and bind in a specific region, then they will emit the radiation that will be absorbed into the microscopy. That is autoradiography, also done in the electron microscopy. So this localization of cell constituent, localization of enzymes, autoradiography, they are specifically to observe the parts of the specific parts of the cell, parts or chemicals of the cell. Okay. Now, next move to the staining techniques. Okay, just I'll see the timings. Okay, okay. Staining techniques. So staining, it is called as the imparting color to the microorganism. So staining, how to define the staining is impart color, add color to the microorganism will be called as the staining. Okay, so the staining used, what are all the stains used in the stain? They are classified into the basic stain, acidic stain, neutral stain. Basic stain is cationic in nature, positively charged, anionic stain, negatively charged, and neutral stain, no charge. Example for the basic stains given here: methylene blue, crystal violet, malachite green, basic carbol fuchsin. Carbol fuchsin, commonly speaking, carbol fuchsin, that is basic stain only, and you can call it as basic carbol fuchsin also. Saffronin, these are all the basic stain, positively charged. Acidic stain, okay, eosin, acidic carbol fuchsin, Bengal rose or rose Bengal, Congo red, Indian ink, nigrosin. Neutral stain, complex of the basic stain as well as the acidic stain, and a salt formation because of the combination of cationic and anionic stain, that is called as the neutral stain. Neutral stain. Example is eosinate of methylene blue. Another example is G-SMA stain, that is used to identify the Rickettsia. G-SMA stain used to identify the specifically Ricketts or Rickettsia. Now, the staining method, what all the staining methods are there? There are two types of staining methods are there: simple staining, differential staining. So simple staining, if only one dye or one stain is used, that is simple staining. Differential staining, more than one dye is used. Okay, so more than one stain is used, minimum two stains are used. That's called as a differential staining. Simple staining, imparting color to the parts of the organism, but at a time you can give part only to the one part. Okay, only one part of the organism can be given color. No two parts cannot be stained. So if you are giving color to the cell wall, only cell wall can be stained. If you are giving color to the nucleus, only nucleus can be stained. The color will not attach with the any other part of the cell. So only one part, one stain can be stained at a time. It is used for the mainly used for the morphology of the organism. So morphological studies can be done. Then differential staining. Okay, so this differential staining used to differentiate the organism. So the microbial world, okay, so will be differentiated by using the differential staining. For example, gram staining differentiate the organism into gram positive organism, gram negative organism. Then acid fast staining differentiate the organism into acid fast bacteria and non-acid fast bacteria. So differential staining, okay, used to differentiate the organism. And this differential staining also used to differentiate the parts of the organism. So for example, cell wall can be given with the one color, blue color, and then spore capsule can be given with the another color. So cell wall pink color, then spore can be given, capsule can be given with the malachite green color. So cell wall can be given with the blue color, then spore can be given with the green color. So at a time, two stains, two different parts of the cells are colored and differentiate, okay, how the structure of the endospore will be there inside the cell, how the structure of the capsule will be there as compared to the cell wall. So differentiating the two parts of the cell. So commonly speaking, differential staining, either it is differentiate the organism or it differentiate the parts of the organism. So that are called as the differential staining. Now we'll see about the simple staining. In the simple staining, positive staining is there, negative staining is there. Positive staining, mostly cell wall of the organisms are negatively charged. Cell wall of the organism is negatively charged. If you add the positively charged or cationic, okay, going back, just a minute. Just a minute. So if you use the cationic or basic stains, methylene blue, crystal violet, malachite green, carbol fuchsin, saffronin, okay, all will have the capability to attach with the cell wall because they are positively charged. The cell wall is negatively charged. So if you use the cationic stains, they'll give color to the cell wall. Okay, so that is we are giving color to the cell, that is positive staining. And if you use the anionic stains, that is negatively charged stains like Indian ink, nigrosin, Congo red, they cannot attach or penetrate into the cell. They'll be present outside the cell only. So the cell will be colorless or it will be present in its own color. Normally cells will be transparent in nature. Cells will be here, black color image I generated, but they are transparent in nature. So the microbe will be transparent in nature, surrounding will be colored. That is called as the negative staining. So positive staining, imparting color to the microorganism or cell wall. Okay, the surrounding will be colorless. Surrounding region will be colorless. Cell will be having the color. That is positive staining. Negative staining, the surrounding will be colorful, cell will be colorless. That's why it is called as the negative staining. Okay, here anionic stains will be used. Then coming back to the gram staining, so here gram staining, which uses the four nutrients, so four chemicals for the gram staining they are using. So gram staining invented by the scientist called Christian Gram. So after his invention, the staining technique is named as the gram staining technique. So he uses four chemicals, that is crystal violet. Crystal violet he used, then iodine solution he used, then alcohol he used, then saffronin he used. Okay, so microbes. Okay, so gram staining, how it is done? The procedure it is explained in this region. Explained here. So first we have to add the crystal violet. Okay, that is positively charged. As I told that microbes cell wall is negatively charged. So this crystal violet is called as the primary stain. So after adding crystal violet, you have to wait for 1 minute. Okay, after 1 minute, water wash has to be done. So that the all the cells, okay, it may be a gram positive cells, gram negative cells. So if you are taking cell and adding crystal violet, so all the cells will take the crystal violet. Why? Because they are negatively charged. So they will take the crystal violet inside the cell. All will appear in the violet color, crystal violet color. Then we will be adding the iodine solution that is called as the mordant. Okay, so the gram staining technique, which is primary stain, which is mordant, which is decolorizing agent, which is counter stain or secondary stain. They may be asking question in the GPAT. So primary staining is stain is ultraviolet, mordant is iodine solution, and 95% alcohol or acetone will be used as the decolorizing agent. Saffronin, that is in the pink color, used as the counter stain. So we were seeing the second step. So first step, adding crystal violet, waiting for 1 minute, then water wash, then add the iodine solution. This iodine will firmly fixes the crystal violet very well inside the cell. Okay, so you have to wait for 1 minute, then water wash, then we have to add that 95% of alcohol or acetone. Okay, drop wise we have to add. So only 5 to 10 drops we have to add, so that the color of the crystal violet will go off. Okay, then we have to wash with water. Okay, then we have to add the saffron in. Okay, then we have to wait for 45 seconds. Then we have to absorb under the oil immersion. So how this will be happening is, in the gram positive bacteria, when you add the crystal violet, it will take up the crystal violet and appear in the crystal violet color. If you add the iodine, the crystal violet iodine complex will formed, that will fixes the color very well. If you do the decolorization, the crystal violet iodine complex will not come out from the microorganism, will be permanently present inside the cell. Alcohol cannot remove crystal violet iodine complex due to thicker peptidoglycan and lower lipid content. So why the gram positive organism retain the gram positive organism retain the gram crystal violet color inside? Reason is that the gram positive organism have the high amount of peptidoglycan, thicker peptidoglycan, and low lipid content. Two reasons. Okay, since thicker peptidoglycan is there, okay, the CVI complex cannot escape. This is the one argument by the scientist. One group of scientists are telling that thick peptidoglycan layer is there, so crystal violet iron complex cannot escape from the by the alcohol treatment from the cell of gram positive organism. So decolorization will not remove the crystal violet color in the gram positive. If you add the saffronin cannot enter because already one positively charged crystal violet is present. So saffronin pink color cannot enter inside the cell of the gram positive organism. So gram positive organism appear in the crystal violet color. Another group of scientists are telling that the crystal violet the microorganism contain the gram positive microorganism contain the lower lipid. So this lower lipid content during decolorization process, less amount of lipid is removed. So hole generated in the hole generated in the cell wall of the gram positive organism very lesser or minute. Through this, the CVA complex cannot escape. But in case of gram negative bacteria, lipid content is higher. So alcohol will dissolve the lipid. So more lipid is lost. Big holes are generated. CVA complex will come out. So adding saffronin will be taken up by the gram negative organism. Will appear in the pink color. Another group of scientists, thin layer of peptidoglycan layer is there. So easily the CVA complex will come out. So gram staining technique, okay. So procedure is that crystal violet addition, okay, 1 minute waiting time, water wash, iodine solution that is mordant, 1 minute, water wash, then 95% of alcohol, adding dropwise 5 to 10 drops over the microorganism or smear, that is called as decolorizing agent. Okay, so we cannot add more amount of alcohol also because that will remove completely. Okay, okay, that results will not come. Then only 5 to 10, then water washing, then we have to add the saffron in, wait for 45 minutes. Saffron is counter stain, that is called as the gram staining procedure, and observe under the oil immersion 100x. Then in the principle is that grams positive organism, because of the one principle, because of the thicker peptidoglycan layer, CVA complex cannot come out, saffronin cannot enter because already CVA complex is present. That is one argument. The shortly speaking, in the gram negative peptidoglycan concept is that thin, okay, thin peptidoglycan layer. So CVA complex will be escaped during the decolorization process. So there is no color inside the cell. Saffron can enter, give the pink color to the gram negative organism. So gram negative organism pink in color, gram positive organism finally crystal violet in color. Okay, this point you have to note in the GPAT by exam point of view. Then another concept is that less amount of lipid inside the cell wall. In the cell wall, less amount of lipid. So alcohol will dissolve lipid. So small holes are generated. Small pores are generated. So CVA complex cannot escape in the gram positive organism. In terms of gram negative organism, high amount of lipid is present. So alcohol will remove the lipid, okay, dissolve the lipid. So more lipid lost, big holes are generated. So that will result that CVA can escape from the gram negative cell. Again, saffron will enter, will appear in the pink color. So this is the gram staining principle told by the two group of scientists. Both are mutually exclusive. Mutually exclusive means just like that you are going to a shop, okay, two shirts you are liking, you are taking two shirts. Both you cannot avoid. You are having money, so both are correct. Okay, so both are correct. They're mutually exclusive. You can't omit anything at all. So Just like that, not only sometime if we have to take the decision, both the things are correct means we have to tell that both are correct. So one group of scientists telling about the peptidoglycan concept, another group of scientists telling about the lipid concept. Since both are right, both are looking correct, so they are telling that both are mutually exclusive, both are correct. They are telling like that. Okay, so the concept or principle behind the gram staining. Okay, in two lines you can tell that alcohol cannot remove CVA complex due to thicker peptidoglycan or lower lipid content in the gram positive. Alcohol remove CVA complex because thinner peptidoglycan, high lipid content, lipid dissolve in the alcohol so that big holes are generated, CVA complex will come out. So this is the logic in the gram negative bacteria which loses the pink color, which loses the primary stain and take up the saffronin. Okay. Then next, acid fast staining. It is also called as the Gill-Nielsen staining. Okay. So here only difference is that carbon used as the primary stain and waiting time is 5 minutes. Heat also applied. So we have to add the carbon or the microorganism. The procedure I am explaining first. We have to add carbon then heat applied for 60 degrees Celsius. Vapors will start to generate, then stop the heating, then keep aside the slide for 5 minutes. Water wash it. And second step, add 3% of hydrochloric acid or 3% of sulfuric acid. Here OF is there, it is R, OR. Okay, I'll change the little mistake and upload in the website so that you can download it. Small mistakes are there, I'll change it here. 3% of HCl ROR. Okay, HCl లేకపోతే sulfuric acid along with 95% of alcohol has to be added. This is used as the decolorizing agent. Wait for 5 minutes. Then after that water wash, then we can add methylene blue or malachite green we can add. So here I have taken as the methylene blue example. Okay, so acid fast bacteria, mycobacterium species. Okay, mycobacterium tuberculosis, mycobacterium leprae. Okay, this organism causing the tuberculosis and leprae, all of you know that. Okay, so mycobacterium species are called as the acid fast bacteria. So this mycobacterium species can be differentiated from the other bacteria by using this method. Okay, so here I have only mycobacterium species are acid fast bacteria, remaining all microorganisms are non-acid fast bacteria. Okay, so how this is happening is mycolic acid is present in the mycobacterium species. This mycolic acid is heat resistant. Okay, this mycolic acid is a lipid which is highly heat resistant and highly acid resistant also. So if you treat with acid and alcohol, mycolic acid will not dissolve. So this carbonfusion cannot come out from the acid fast bacteria or mycobacterium species. So once you treat the cell acid fast bacteria of the carbonfusion will enter into the cell. If you are decolorizing with the acid and alcohol also, this mycolic acid will not dissolve, so carbonfusion will not come out. Then if you add methylene blue cannot enter. Malachite green, that is also positively charged, that cannot enter. Already crystal violet is present. So the not crystal violet, carbonfusion is present. So the acid fast bacteria retain the carbonfusion, appear in the pink color. Do remember, appear in the pink color. The uh, uh, principle behind the acid fast staining or Gill-Nielsen staining is presence of mycolic acid, which will not be dissolved easily by the acid treatment, so that carbonfusion or primary stain cannot be removed. So it is present inside the cell. Okay. So the secondary strain cannot penetrate. So non-acid fast bacteria, there is no mycolic acid. So acid and alcohol treatment remove the carbonyl fusion. Then methylene blue treatment, methylene blue will enter, cell will appear in the blue color. If you use the malachite green will enter, cell will appear in the blue color. So secondary stain, you can use the methylene blue or malachite green. So this is acid fast staining, Gill-Nielsen staining. This is also one of the main important question. So in the previous slide, it completely covers the procedure, waiting time, and principle of the gram staining. This slide covers the acid fast staining procedure, waiting time for the every step, then finally principle also, along with what color that respective organism will appear. So that is also present in this slide. So we just memorize this in terms of the GPAT examination. Okay, because questions are coming. So do remember that only mycobacterium species. So only mycobacterium species respond to the Gill-Nielsen staining. That is acid fast staining. This is acid fast positive bacteria, mycobacterium. Remaining acid fast negative bacteria or non-acid fast bacteria. So, for example, disease identification. So someone is having the cough, tuberculosis. Okay, so directly the organisms are isolated from the sputum. Okay, sputum of the patients. Then if you perform the acid fast staining, if it is shows the pink color or carbonyl fusion is retained, you can tell that mycobacterium. There is no other mycobacterium tuberculosis infection will be there, or mycobacterium will be there. You can tell that clearly. So in some cases, the acid fast stain, okay, used to identify the mycobacterium species, mycobacterium leprae, leprosy diseases, as well as in the tuberculosis diseases. Then next slide. So differential staining. So above acid fast staining as well as the gram staining. So that differentiate the organisms. Okay. So these staining methods differentiate the parts of the organism. Differential staining differentiate the parts of the microorganism. So here examples has been given: endospore staining, capsule staining, flagella staining, storage granules stainings. So there are different type of storage granules are there in the cell. Metachromatic granules are there, starch granules are there. You know that mainly iodine is used. Okay, PHP granules are there. So PHP is polyhydroxybutyrate. Okay, so endospore staining, Scaffer Fulton method, Dorner method is there. Flagella staining, Leifson stain, capsule staining, malachite green is used, malachite green. Okay. Metachromatic granule staining, Albert staining is used. Starch granule staining in iodine solution or iodine is used. Okay. Then, sorry. Now, gram positive bacteria, gram negative bacteria. Some examples I have given. Cocci. So coccus means the word coccus is coming cocci. Okay. So enterococcus, spherical shape, round in shape, rod. Okay. Okay. Rod shaped. Okay. That is bacillus. Okay. So example for the rod shaped organisms given for the gram positive organism, and Cocci organism also given. So in this, remember sudden organism, Enterococcus. Okay, Enterococcus. I have marked important to remember. So why? Because as some GPAT questions are coming. Okay, so example for the gram positive organism. So the following organisms are all are gram positive except one. Okay, like that GPAT question. There is a chance it may be coming. I'll show the examples to you also. So this slide Okay, so next 4 slides, you try to remember that very carefully. So gram-positive organism, Enterococcus, Peptostreptococcus, Staphylococcus, Streptococcus. So carcass mainly, cocci, gram-positive one. Gram-positive rods, Bacillus, Clostridium, Cornybacterium, Lactobacillus, Listeria, Propionibacterium. In this, mainly you have to remember this, I have marked in the orange that you remember it. Okay, memory. You have to memorize. The next slide, okay, which organism in the gram-positive bacteria aerobic, okay, require oxygen for their growth, anaerobic, which is not required oxygen for their growth, okay, rods, okay, that is uh, rods, okay, bacillus shape. So which is required aerobic conditions, which is required anaerobic conditions. So I have given an example: aerobic staphylococci, streptococci, enterococci. Okay, aerobic, aerobic in the rod, bacillus, listeria, nocardia. Anaerobic, artidomyces, clostridium. So if I go to the previous slide, okay, in this here, Clostridium only anaerobic, remaining all are aerobic only. Propionibacterium aerobic, Listeria aerobic, Lactobacillus present in the milk aerobic, then Erysipelotrix aerobic, Cornybacterium, okay, diphtheria aerobic, Bacillus, all the Bacillus aerobic, Clostridium, okay, anaerobic, Clostridium species anaerobic, Clostridium tetani, okay, Clostridium botulinum, okay, botulinum, okay, sorry, Clostridium botulinum, Clostridium botulinum, Clostridium pyrogenes. Okay, we'll see later while seeing about the organism and diseases we'll be seeing. Okay, so you remember which are all aerobic, which are all anaerobic. Anaerobic rod Clostridium you keep it. Anaerobic Peptostreptococci you remember in the gram positive. Remaining all are gram positive aerobic organisms. Anaerobic Clostridium, Actinomyces, remember. Remaining all rods are aerobic. Here also aerobic, only Peptostreptococci, remaining all are aerobic. Anaerobic is Peptostreptococci. Do remember that. Okay. Then next is that gram negative cocci, Acinetobacter, Neisseria gonorrhea, important organism. Non-enteric rod. Enteric rod means enteric organism present inside the gastrointestinal tract. Enteric organisms are present in the gastrointestinal tract of the human being and animals. They are enteric rods, non-enteric rods. So enteric organism, okay, enteric rod organism, okay, all are gram negative. Okay, only Enterococcus, that is gram positive. Enterococcus, gram positive. Okay, Enterococci, aerobic. Okay, Enterobacter species, E. coli, Escherichia coli, Helicobacter, Klebsiella pneumoniae, Proteus vulgaris, Pseudomonas aeruginosa, Salmonella typhi, Shigella, Vibrio, Yersinia. Okay, these organisms are important enteric rod organism, comes under the gram negative. Non-enteric Bordella, Brucella, Haemophilus, Pseudomonas. So this you remember. Okay, Pseudomonas some species. Okay, Cocci, Acinetobacter, Neisseria. You remember. Then anaerobic organism. So all organism I have, okay, so mentioned there, okay, I have mentioned in the previous. So most of them are all are enteric, all are aerobic in nature. All organisms are aerobic. Okay, so here also most of them are aerobic. Okay, Neisseria aerobic. Neisseria aerobic. Okay. Villorella anaerobic in the cocci. Then bacteria, fusobacterium anaerobic. Remaining all you keep in the aerobic only. Anaerobic, bacteria, fusobacterium. Okay. Now miscellaneous acid fast bacteria, mycobacterium. I already told that. Okay. Intracellular bacteria, chlamydia, rickettsia, and borrelia. Okay. You poorly staining bacteria, mycoplasma, legionella, helicobacter. This you have to remember. Okay. So nocardia modified, rDNA modified nocardia, they comes under the acid fast. Okay, that won't come in the GPAT question. Acid fast bacteria means, do remember mycobacterium. Now, we'll move to the model GPAT question in the previous year. The phase contrast microscopy is valuable in studying the living cells. Okay, phase contrast microscopy valuable in studying the living cells. Living cells, which are living cells are unstained. Stained means killed cells only stained. Fluorescent antibody, fluorescent dye if you are using, that is also you have to organism will die. Treat with a fluorescent dye, organism will die. So unstained living organism. So C is the right answer. This is one of the question came in the old GPAT question. So I collected GPAT question of 1998 to 2019. So the portions which I covered today. So which are all the question present in the old question that I compiled, I have given. For your future reference, I didn't mark the answers at all. Now I am telling answers here. Okay. Later on, after studying the slides, you can also practice this question in your home lonely. Okay. For microbiology and biotechnology, end of session, every last 15 minutes, I'll discuss the question of the previous year question paper. Answers I'll give in the last day only, but I'll tell the answers today. But printed answer I'll give in the last day only. So phase contrast microscopy is valuable in studying living cells which are stained, unstained. C is the right answer. Then which of the following are obligatory intracellular parasites? So they can live only inside the cell only. So virus, then rickettsiae. Rickets we didn't see. Virus and rickets. A and D as the right answer. Okay, A and D. Fungus can be grown in the media. All will not come. So fungus is not intracellular. All will not come. A, B, C, D, all will not come. A, B, C also not coming. A, B, C, because fungus is a fungus can be grown in the media. C and D, C and D here A is missing. So C mycobacterium is not a intracellular. It's not a intracellular. Okay. C and D virus is missing. So A and D, A virus intracellular, rickets D intracellular. So A and D is the right answer. So now while answering for the GPAT question, sometimes you have to omit the answer, then choose the right answer. Then okay. Then another question, major difference between the prokaryotic and eukaryotic protein synthesis mechanism are in which part of the process? So protein synthesis flow, which part of the process prokaryotic, eukaryotic difference is there? In the initiation of synthesis, the chain termination process, in chain elongation process, none of the above. So I told that prokaryotic, first amino acid added in the protein, that is formyl methionine. Formyl methionine. In the eukaryotic, methionine will be added. So, in the initiation of the synthesis, initiation of the protein synthesis, okay, so there is a difference between the prokaryotic and eukaryotic. That's not there in the chain termination, not there in the chain elongation. So all will be same, all process will be same. Starting only there is a difference between the prokaryotic protein synthesis and eukaryotic protein synthesis. So that is A is the right answer. Then next question. Which of the following pairs is mismatched. Aerobic helical bacteria gram negative, mycobacteria acid fast bacteria. So C is not mismatched. Mycobacteria is acid fast bacteria. Enteric bacteria, gram negative bacteria. As I told that, enteric rods. So enteric rods means which is living in the gastrointestinal tract. So in our body, they are gram negative bacteria. Enteric bacteria, gram negative. So C and B, okay, that is not there. Pseudomonas, that is gram positive bacteria. That is actually Pseudomonas is a gram negative bacteria. Pseudomonas is mentioned in the gram positive. This is the wrong pair. D is the answer. So if you see here, Pseudomonas, okay, I'll show my previous slides, okay. So Pseudomonas, gram negative bacteria. Pseudomonas comes under the pseudo gram negative bacteria, okay. Pseudomonas, gram negative bacteria. Okay, so Pseudomonas gram negative, here it is given as the gram positive bacteria. So aerobic helical bacteria gram negative, that is correct only. Okay, so mycobacterium acid fast correct only. Enteric bacteria gram negative correct only. Pseudomonas gram positive. So these two compulsorily you will be knowing. If you are having two doubts, that time also, if you know better, if you studied all the gram positive, gram negative organism clearly, then you can answer this. Otherwise, be difficult. Then all the following are gram negative rods. All are gram negative except gram negative rod. Okay, so E. coli gram negative, okay, Salmonella gram negative, Shigella gram negative rod, Clostridium, okay, so gram positive organism. Clostridium is a gram positive organism. So you see in the previous slide. So Clostridium, gram positive organism, okay, Clostridium, this is Clostridium, this is gram positive organism. So see here, okay, gram negative rod, Enterobacter is there, okay, E. coli is there, Salmonella is there, Shigella is there. That's why I have marked some important bacteria, so you do remember, okay. Okay, Clostridium, gram positive. Here, all the following are gram negative rods except. So A is the answer. Then here, disease in gram negative diplococcus associated with the urinary tract infection, okay, pelvic inflammatory diseases, conjunctivitis, meningitis, okay. So gram negative diplococci, Neisseria gonorrhea, okay, gram negative, okay. So streptococcus gram positive, hemophilus gram positive, chlamydia, okay, gram positive, Neisseria gonorrhea is a gram negative. So by using gram negative only you can identify the organism. So later on, urinary tract infection, gonorrhea only causes. So we will see the diseases. Okay, that time you can betterly understand. But this question you can answer. Okay, so without only by using the clue a gram negative. So if you see the slide, okay, I'll show you the slide. Chlamydia, okay, it's not coming in the gram positive one, gram negative, chlamydia, gram positive or gram negative, it is a different intracellular bacteria, okay. So coming back, okay, Neisseria, gram negative, okay. So remaining streptococcus, okay, streptococcus, okay, so clostridium, another, okay, bacillus, enterococcus, they are all the gram positive bacteria. So they have given, okay, you see this question, you refer this, okay, I'll tell you. Okay, gram negative. Okay, chlamydia, not in the gram negative, gram positive. Streptococcus, it is also in the gram positive. Hemophilus, gram positive. So Neisseria gonorrhea is the answer. Then, sorry. Okay, saffron is used as a reagent to detect gram-negative bacteria. A. Gram positive acid fast Mixozoa, gram-negative bacteria. Then a specimen obtained from the patient's cerebral fluid, cultured in the specialized media for about 5 weeks, showed the presence of bentrax and tested positive with Gill-Nielsen reagent. Gill-Nielsen reagent, acid fast staining, straight away you click that Mycobacterium tuberculosis. Mycobacterium word is there, click it. Don't see any other, you don't know the diseases, you don't know other things. Gill-Nielsen reagent, positive organism, Mycobacterium tuberculosis, Mycobacterium species. That's all. So, like the way you can answer the question, no need to see the others at all. Okay. Now, today I'll complete the session. Okay. And tomorrow I will remaining will be seeing slowly. Then answers also. Okay. Questions also will discuss. Lastly, answer the fourth day I'll give the answers. Okay. So thank you for your patience. Okay. So I know it's very hard for the student to listen more than 2 hours, but those who are listening, I can understand your the kind of what you said motivation towards that GPAT. So because of that, you was sitting for more than 2 hours. So my efforts, I feel, definitely will satisfy you. And if any comments about the today's session, if it is not good, if it is confusing, some doubts are there, you just put it in the query so that I can clarify tomorrow. Okay. And thank you. And we'll see you tomorrow. Thank you. Thanks a lot. Hello? Hello? Organizer? Organizer? Organizer?
Sir, okay, sir. Okay, sir.